Welcome back. Before we learn about how mRNA can be used as a medicine, let's talk about some of the properties generally required of medicines so that we can have control over their effects and ensure their safety. Please take a moment to review the learning objectives for this lesson. An ideal medicine has three basic attributes. 1, its effect is dose-dependent. 2, its effect is time-limited. And 3, it can be dosed repeatedly. Both classic small molecule and protein medicines have all three of these properties. A limited duration of effect is particularly important because if one experiences side effects, you can simply stop taking the medicine. Let's check to see whether mRNA has the right features to be an ideal medicine. Unlike DNA, which serves as the permanent repository of your genetic information, both proteins and mRNAs are inherently transient. With very few exceptions, most proteins are under constant flux. They have limited lifetimes and must be regularly replenished. Likewise, the instructions for making them, the mRNAs, are constantly made and destroyed, only to be remade again. This enables your body to rapidly change the amounts of key proteins so that you can respond to changing conditions. To illustrate this, let's consider your circadian clock. This is the timer in your body that tells you when to be active and when to rest. The proteins responsible for the clock are aptly named clock proteins, and they appear and disappear with astounding regularity every 24 hours. Your body accomplishes this with a daily cycle of clock mRNA creation and destruction. The amount of protein produced at any one time is tightly tied to the amount of mRNA present at that time. Over and over, for every day of your life, your cells get their daily dose of clock mRNAs, driving expression of clock proteins. Although all proteins and mRNAs are transient, each different protein and its corresponding mRNA has been tuned evolutionarily to have an optimal lifetime. Now, let's learn about a way to measure how long proteins and mRNAs stick around. A common measure used to describe how long a particular substance sticks around is its half-life. Half-life is the amount of time required for half of a substance to disappear after it has been made. Structural proteins tend to last the longest. The protein crystalline in the lens of your eye has a half-life of more than 70 years. Once incorporated into the lens, crystalline cannot be replaced and needs to last you your entire lifetime. But this is highly unusual. Antibodies are among the longest-lived proteins, but their half-lives are only 10 to 21 days. Remember that huge protein Titin? Its half-life is just 3 days. Shorter still are the half-lives of proteins that mediate metabolic reactions. Remember that metabolic chart from Module 1? An example from this class is the protein glucose 6-phosphatase, the one that's missing in von Gurke disease. Its half-life is somewhere between 8 and 48 hours. At the very short end are signaling proteins like the hormone relaxin, whose half-life is less than 10 minutes. So protein half-lives can span from minutes to decades. The median protein half-life is estimated to be about 30 hours, or just over a day. So what about mRNAs? How long do they last? mRNAs are not nearly as long-lived as proteins. The typical human mRNA has a half-life of just 5 hours. Of course, like proteins, mRNA half-lives do cover a range, with the shortest being just a few minutes and the longest being about 5 days. Importantly, no mRNA lasts for your entire life. They are short-lived by design so that your body can make the appropriate proteins in the appropriate amounts as your body requires them. Because, 1, mRNA is transient. 2, there is a dose dependency between an mRNA and the protein it encodes. And 3, an mRNA can be induced over and over again. mRNA has all three properties of a classic medicine. In addition to these, there are 3 more key properties of mRNA that make it extremely attractive as a new way of making medicines. We learned about the first of these in the previous lesson. That is, mRNA has the ability to direct the body to make any type of protein, not just proteins that do their job outside of cells. So mRNA medicines have the potential to meet many currently unmet medical needs. These range from rapid development of vaccines against new pathogens as quickly as they emerge, to stimulating the immune system to fight cancer, to treating patients with inborn metabolic errors by giving them back the ability to make the protein they're missing. The second feature is the small manufacturing infrastructure requirements for making mRNA medicines. Unlike recombinant proteins made by tissue culture cells grown in huge bioreactors, the process of making mRNA medicines has a much smaller manufacturing footprint. mRNA medicines are made using solely chemical, biochemical, and biophysical processes that do not require mammalian cell culture. The equipment for these processes is relatively small and can be mobile, making the manufacturing facility highly adaptable. Because the infrastructure and equipment required to make mRNA medicines are relatively modest, manufacturing sites can be easily set up in multiple locations around the world. And because the sequence of nucleotides that make up an mRNA is simply information, when mRNA drug designers have successfully created a new mRNA medicine, say a vaccine against a rapidly emerging pathogen, that mRNA sequence can be instantly delivered digitally to every manufacturing site for local synthesis and distribution. In the future, as technology improves, it might even be possible to miniaturize the entire manufacturing process to take place in a single benchtop machine. The third feature is that unlike small molecules and proteins, mRNA is an information-containing molecule. It is not itself the active medicine. mRNA is simply a recipe that tells the body how to make its own medicinal proteins. Once a drug maker knows how to make and formulate clinical-grade mRNA and how to direct it to the desired cell type, for example, immune cells for vaccines or liver cells for a metabolic disorder therapeutic, they can simply swap out the previous mRNA sequence with a new one to create a new mRNA medicine of the same type, for example, a new vaccine or a new therapeutic. mRNA medicines are therefore a platform technology. According to Techipedia.com, a platform is a group of technologies that are used as a base upon which other applications, processes, or technologies are developed. These days, most of us have a platform technology in our pockets, our smartphones. The phone's hardware and operating system constitute the platform. This platform then enables rapid development of countless apps that we all use every day. In the case of mRNA medicines, the hardware is the manufacturing equipment used to make the mRNA medicines. The operating system consists of the processes and know-how for making mRNA medicines at various scales. Importantly, all mRNA medicines of a particular type use the same raw materials and manufacturing processes. Thus, the need for re-engineering to create a new medicine is minimal, and this can greatly accelerate the timelines to develop the apps, that is, the individual mRNA medicines. Yet another advantage of mRNA medicines is that they can be easily multiplexed, meaning that more than one mRNA can be delivered in a single medicine. This can provide the body with instructions to make multiple different proteins at the same time. Remember that some proteins act as multi-protein complexes. If a particular application requires synthesis of the entire complex, the mRNAs to encode each component of the complex can be delivered simultaneously. Alternatively, mRNA vaccines encoding proteins from different pathogens can be combined into a single shot so that you can be vaccinated against several different infectious agents at the same time. Now that we've learned about the potential advantages of mRNA medicines, in the next lesson we'll learn about the many challenges that had to be overcome to fulfill this promise.